Moment, let the recording start. Yeah, so we will be continuing on the sequencing technologies, whatever are there in place. We'll start from the first generation sequencing, then the second generation and the third generation. Second generation is also known as the next generation. So we'll see what those things are. So, but before we left in the last session, we talked about restriction enzymes that are methylation sensitive and those which are methylation specific. These two things we talked about, right? Do you remember?
Yes, sir.
Okay, so today we'll be talking about bisulfite conversion, one of the most used ways for methylation specific analysis. So here essentially we treat the DNA with bisulfite, sodium bisulfite, and it was discovered in 1970s. So what it does, it converts unmethylated cytosine residues to uracil in a single stranded DNA. While 5-methyl cytosine, the methylated ones, they do not change. So this is how it works. So whenever there is a methylated cytosine, it does not change on bisulfite treatment. But if there is unmethylated cytosine, let's say here, then it changes to uracil. So by this way, we can actually compare whether which cytosines are, let's say, methylated and which ones are not. So why are we, not using restriction enzymes which are methylation specific or sensitive, but instead doing bisulfite conversion? Anybody could guess why is that?
Because if we means restriction enzymes, if methylation is not present, then means for methylation sensitive restriction enzymes. So it will cut the DNA, and hence we cannot compare with the reference genome. That will be a bit difficult of a task.
So
So
essentially,
this
some of the cytosines might get missed because
might get missed.
if the methylation is not present in the restriction recognition site, essentially. So that is the answer. You're right. So this is how bisulfite conversion happens. It happens through a sulfonation reaction, very similar to what we have seen deamination. Cytosine converts to uracil. That happens also in our body. So this amine group is removed due to bisulfite treatment. So that is what happens. And then what are the pros? The positional methylation information we can get in a single base resolution, right? Which particular base has been methylated, which is not, we can get that kind of information. While previously we were not able to get such resolution in terms of information. What the disadvantages are that the DNA gets degraded because the process involves, change in pH, higher temperature, and incubation time. So at that, in these conditions, DNA they starts getting fragmented. So we get a fragmented version of DNA with around 150 to 200 bases. So if we have, let's say, a very big DNA, like in case of humans, then that DNA, if it gets broken down into such small fragments, it will be very difficult to analyze them. So this is how bisulfite conversion works. Now I'll talk very briefly about Sanger sequencing. Do you know what it is? You'll be reading this in RTT.
No, sir.
So can you see
Sir,
this is
heard about it.
Yes, yes. So it's actually even now, even today, it's a gold standard for sequencing. It's one of the most cheapest ways of sequencing. And the principle is very, very simple. So what essentially happens, this is a DNA molecule. Can you see this?
Yes.
This has a 3' OH, which will extend the DNA will get extended using the 3' OH, right? During 5' to 3' polymerization.
Yes, sir.
Okay, now what if I do not have an OH group here? Instead, I have H group. So such kind of dinucleotides will be called uh, deoxy di, dideoxynucleotide triphosphate, DDNTP. So both the, I mean, if there is a OH here, then it will be a, what will it be?
Sir, please repeat the question.
In a ribose sugar, if this is the sugar, if there are two OH, then what will it be? Which kind of molecule?
That will be RNA,
RNA, but
means
it will be
ribose sugar.
ribose.
Ribose
If
sugar.
we if I remove this O, then it will be.
Deoxyribose.
If I again remove this O, then it will be dideoxy.
Deoxyribose.
Yes, correct.
We
So
did this
what?
in molecular biology lab.
Yes. So what happens is if we have a dideoxy nucleotide, what would happen? Will it
Uh, Addition of nucleotides will stop.
is exactly that is the point. So these dideoxy nucleotides, these are fluorescent labeled. Fine, that is clear, and there are normal DNTPs also. So we will go under it will this will undergo a normal polymerization reaction, and each base will be added and it will stop. If DDNTPs are added, it will stop. Clear?
Yes, sir.
If DNTPs are added, then it will continue until a DDNTP is added. So we will get multiple fragments of varying length. If we uh, separate these fragments in a gel, so we'll get different lengths, and when it will be detected, this fluorescent signal will be detected, then we will get the sequence of the DNA. Is that clear or is it not?
Yes, sir.
Yes, sir, clear.
So you will be reading this again in your I mean, not molecular biology, RDT essentially. You'll be reading this again. So what happens? Let's say, so here if A is added, then this will have a separate fluorescence. G will have a separate fluorescence. By that we will have a chromatogram. This is how the result looks like, and using that we essentially decide that what are the bases which are present. Fine. So this is Sanger sequencing. It is good for small amount of DNA. It is good for that, but if you go for very long amounts of DNA, like say human DNA, then it takes a lot. It will take a lot time. So that's a problem. So
Sir, what is a chromatogram? Sir?
chromatogram is nothing. It's essentially all the fluorescent labels. Since you have added all the fluorescence measurements, they will be present as chromatograms. So these all you can see the spikes.
Yes, sir.
These are individual nucleotides. So let's say A has a separate spike, G has a separate spike. So using that, sometimes in some regions, the signal is not good. So we manually inspect the chromatogram and see whether the reading is correct or not. So that's why
And sir,
chromatogram
what
is
is the
important.
significance of that baseline? Like the line which is below means the amount is less at that point. The
Okay,
others
so
are
there
not
are
present.
some DD and TPs are already present in the mixture, so they will also give some fluorescence, isn't it?
Okay, okay.
So those kind of some noise is always present in these kind of things. Whenever you go for spectrophotometry also, you'll find some noise. So anyway, we'll talk about methylation specific PCR. So we design specific primers to test whether methylation is present or not present on bisulfite converted DNA. If you remember bisulfite conversion. So we use two sets of primers, and it is used to target, analyze methylation in the target promoter regions or something like that. So this is how it works. So let's say this is a promoter region of two. There are two alleles. One is methylated, another one is not methylated. So if it is methylated, there will be no change in Cs. But if it is unmethylated, it will be converted to uracils using bisulfite treatment. You know this. So then we can run PCR specific to both of them and see where we get the band. So this way we can analyze whether the DNA or the promoter of interest was methylated or unmethylated. Clear?
Yes, sir.
Okay. So next, let's move forward. Then, okay, another kind of problem which can happen is that these DNA, what will happen if you try to run PCR on methylated DNA? We have discussed this previously. Hello, any answers?
Wait, sir. Thinking. Couldn't remember actually. Wait.
Let's say if I run PCR on this, what would happen? Will the methylation remain in the product?
Oh no, no. The methylation will not remain in the product.
Okay, so in what way can you actually concentrate or get a higher amount of methylated DNA in specific? What would be one of the ways of doing that? Anybody would like to suggest something?
Like we will add some methylase enzymes.
Methylase will not.
No, that will
Methylase,
not.
even if you add it, it will go on methylating everything. So will it
It
help?
will go on methylation.
Mayuk, anything? I think you want to answer.
No, sir. Actually, I was getting a bit confused. We cannot add. Can we add methylase enzymes externally to a DNA? So that
So
it
we
gets.
cannot actually. Not that I know of. I have not heard of this. But anyway, even if we do, the unmethylated cytosines will also get methylated. So that's not solving any problem.
So
Anybody
what's the
else?
question? We need to unmethylate, right?
No, we
We,
don't need to unmethylate. I
then
want to get, let's say if you are taking human DNA, there will be methylated DNA, there will be unmethylated DNA.
hmm.
Most of it will be unmethylated only. I mean, I really do not know the proportion, but let's say there's a lot of unmethylated DNA, there's a very little amount of methylated one. So when you try to do something like this, it will be very problematic, isn't it? Because methylated proportion is very less. So getting hold of it is difficult. So how would you do that? That's the question.
How would we get the methylated one?
We can do bisulfite conversion.
That is correct. But I mean, that's
But it is not quite specific, right?
although it is specific, but if you do it in promoter regions, it's good because those areas are specifically methylated. So have you heard about immunoprecipitation?
Heard the name, but don't know the process.
Yes, sir.
So Eliza heard about it.
So like introducing chemical differences
Yes,
within them, right?
Yes, sir.
ELISA
Elisa,
cells.
we know. We know the procedure name.
What is there in Eliza?
Enzyme linked immunosuppressant. So like
Nice.
we
What
have
is the
Nice.
some
main component?
antibody, linked with the enzyme. So if there is like a specific antigen, no, and then the antigen will interact with that antibody, and the enzyme will react with a particular substrate, so we can actually detect the presence of that antigen there.
Right, correct. You're right.
As
So methylation dependent immunoprecipitation is something we can do. But wherever there is methylated DNA, we can create an antibody which is specific to this methylated DNA. It will bind to this methylated DNA, and then we can cause its precipitation because we will then add another, antibody which will be specific to this antibody, and it will cause that kind of DNA to get precipitated. Is that clear or is it not?
sir, I go repeat.
There will be one antibody which will only bind to the methylated DNA. So that way, if we pull down this antibody, then we will also get the methylated DNA together. So that is actually immunoprecipitation. There is something called co-immunoprecipitation also. Sometimes when two proteins are interacting, let's say this is protein A and this is protein B, and you create an antibody for protein B, and you precipitate this, pull down this antibody, then you will see protein A will also come up because they both
Yes.
of them are interacting. Very similar to that, when we have antibody for only methylated DNA, we can actually pull down these antibodies and we'll get only methylated DNAs, methylated DNA. So that way we can, run PCR, let's say RT-PCR, real-time PCR essentially, and we can compare the CT values. So where will we get a lower CT value if we are checking methylated DNA? In the immunoprecipitated ones or the normal ones which were not
Immuno
immunoprecipitated? It will have higher CT value, lower.
The immunoprecipitated ones will have wait, one minute.
Lower CT values is means higher amount of DNA.
Higher amount of DNA. Acha, lower CT value.
Yeah. So we'll talk about microarrays. Ever heard of it? It actually comes, this kind of questions come in GATE usually. So microarrays, do you know?
No, sir, not yet.
So, okay. So essentially they have, there are probes and there are wells where we detect, there is hybridization of your target DNA, and then we detect the fluorescent signals. What in this context, what happens? So there are fragments of the genomic DNA which are used as probes. So in a well, there will be fragment of DNA which are probes. Fine. And then our target gene of interest, let's say methylated CPGs, they will hybridize with this probe, and these our target, gene or DNA will be fluorescent label beforehand. So in each well, you will get some fluorescent signal if it hybridizes with the probe that you have in the well. So let's see how it looks like. So see, here we have the input DNA. It is, let's say, mostly unmethylated. So we will do fluorescent labeling with one dye. And if we do immunoprecipitation, we enrich the DNA for only the methylated ones. They will label it with another dye. And when we check this, this is how a microarray will look like. So the methylated ones will be in red, the unmethylated ones will be in green. Shinjan, you have any question? Yeah, I'm coming to it. So essentially what happens is, this is how a well looks like. In the right hand side, you can see.
Yes,
Hello.
sir.
Yeah. So in a well, there will be probes. Fine. So these probes are essentially nothing but they are like primers. They are like some DNA fragments which are there, which will bind to your target DNA. Clear?
Yes, sir.
So let's say it you are treating your target DNA with some fluorescence labeling. It is also used for disease detection also. So let's say you have some mutations. Let's not talk about CPGs here. Let's talk about some other disease model. It will be easy for you to understand. So let's say you have some mutation, like you have ACT instead of TGC, something like that. You have some mutation, like this kind of mutation is there. So if you have treated this DNA with one label and this DNA with another label, these are two colored fluorescent labels. This is one way of doing it. Another way is using antibodies also. Like they have used antibodies like you do in ELISA. It's like that. So these antibodies are labeled. Both the ways you can do. Essentially what happens, they will only bind to specific probes based on complementarity.
Yes, sir. These probes are pre-designed, sir.
Yes, these are pre-designed.
Pre-designed probes, okay. And they will bind to specific fragments of
When they
target DNA
bind
which
to the
have
specific
fluorescence.
probes, those wells will show fluorescence based on
Fluorescence,
what.
because
So, so let's say if they show green fluorescence, that means this is the this is how it exists. If they show red fluorescence, then this is how they exist. If there is both green and red, what will be the color? Yellow.
okay, yes, sir.
So this is how essentially microarrays are designed. Disease, it is more helpful for diseases, but let's say for this, I'll give you a good example on this. Let's say you are going to a doctor with a symptom like you have like upper respiratory, some kind of problem, you have respiratory problem, you have cold and cough, those kind of symptoms. So there could be a lot of things which could happen. You could have COVID, you could have some other rhinovirus infection, you could have a lot of, lot of viruses could cause this kind of symptom, right? Clear on that?
Yes, sir.
So if I design a microarray which will have, different probes, different wells will have different probes for different viruses. and I give your sample, and whichever will give me the fluorescence, I know that this is the virus that is infecting you. I mean, is that clear?
Yes, sir.
So some practical examples, but in our context, we are only seeing what CPGs are doing. So if there is methylated, I mean, methylated CPGs, mostly cytosines are methylated. So if they're methylated CPGs, they will give us a separate signal. If we have unmethylated CPGs, it will give us, let's say, green signal here. If we have mix of both, then we'll have partly methylated signals. Now, tell me, if I do not do immunoprecipitation and I have a very less proportion of methylated DNA, so what kind of signal will it be?
Very less methylation, that will be means more green dots will appear then.
Yes, yes. So yeah, those kind of things might happen. So here also, if you want to really check methylation, you need to perform methylation immunoprecipitation. Shinjan, is it clear, or is it still confusing?
Yes, after the example, it's
Yeah, fine, good.
That means
Uh,
microarrays always require immunoprecipitation, right?
no, 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 not. It depends on your context.
On the context. Okay, okay.
So yeah,
So,
not always.
microarrays basically up to like fluorescent labeling and then hybridization. And then to visualize it, we may use immunoprecipitation as one of the methods.
No, no.
Uh,
Immunoprecipitation is done before in during the sample preparation phase, so
during
that
the
you
acha,
get,
acha.
yeah, yeah. Even if you have a very low viral load, you give the entire DNA, it will contain human DNA also. It will contain viral DNA also. So if you do some targeted, let's say PCR, or if you perform, in that case, I think immunoprecipitation is not possible. But you're understanding the problem, right? So if the proportions are not good, and then there will not be a good result essentially. So it's just a visual result. It's not quantitative as such, if I
so
can
just
explain.
one thing. So what
This
does
part is
sonication mean?
this part is a bit
What does
confusing. Wait.
Sonication means that one part I missed.
sonication? I did not explain. Sonication is, giving sound waves. So what would happen if you give sound waves to DNA?
The DNA will break.
Yes, correct.
So then, what's the difference between this and denature? Like both are breaking.
No, no, denature is like denature. What happens is, double-stranded DNA, it becomes a single-stranded DNA.
Oh, so this only breaks the DNA, but keeps
That
it
is
double stranded.
fragmentation. Sonication causes
Acha, acha.
fragmentation.
So, sir, first we are doing suppose sonication, like in simple words,
Also,
we are breaking the
sonication is
DNA.
sometimes done, let me again, sorry to interrupt. Sonication is also done mostly not to actually break DNA, but, this to lyse the cells, to take the DNA out of the cell.
Okay, sir.
Is that clear?
Means it's
Yes.
done to the cell, not the DNA.
DNA also you can do. I mean, that's not a thing, but usually it's not done. There are two types of sonication. One is probe sonication and bath sonication. So probe sonication, I mean, we can go at lengths about it. Probe sonication is very destructive. So, but bath sonication is kind of, it's better because you have a water surface there. So you'll learn about it again. But the thing is, cells are sometimes crushed by mechanical crushing, or you do vortexing sometimes. These kind of things you do. Sonication is an alternative. Yeah, Antik.
Yes, sir.
Yes, sir. So, first in this process, I mean, in microarraying, we first break the DNA. After that, we label the DNA by some fluorescent dyes. Methylation has a like red color upon fluorescence, and methylated will have green color on fluorescence. And if mixtures of both are present, then we will have yellow fluorescence. So then we are adding it in three separate wells, which will have some specific pre-designed probes, which may hybridize with the DNA. Uh,
Actually,
right?
we do not add it in three wells. We
Aha,
actually, all the probes are present in a single well. They'll have
in a
millions
single
of
well.
probes.
Millions of
See
probes.
here, each glass slide, it can have millions of probes. So even if you add it in one well, whichever you know probe it hybridizes with, whichever, I mean your DNA hybridizes with whichever probe, it will give fluorescence, because there is
It will.
washing step involved. If it does not hybridize, then it will be washed away.
Okay. Okay.
Okay, clear. Then next generation sequencing. Let's see, it's very popular nowadays. So probably some of you will be doing this in your, I'm not sure, masters, maybe PhD, I don't know that, but probably you'll be doing it. So we first extract the DNA, then we prepare libraries, and then sequencing is done, and then the analysis step. So step 1 to 3 is one headache. Step 4 is another level of headache. So you will require a lot of computational skills. So for that, so we'll see how next generation sequencing works. But this is the basic workflow, which is common to all next generation sequencing, even third generation sequencing, which we'll be talking about. So clear on that?
Yes, sir.
Okay, let's see. So this is how it works, next generation sequencing. So you will have adapters which will be attached to both the ends of the DNA. Why are they attached? So that they can, if these adapters are present, they can, you know what are adapters and linkers, right?
Yes, sir.
No, so
Okay.
can you once tell?
So adapters are nothing. These are like these are short stretches of DNA. Usually these DNA, this I mean sequence is not present inside your target DNA of interest. So these are some unique DNA patterns which are added with your DNA in both the
Can we
ends.
call them primers?
No, 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 no. I'll
Call
tell
them
you why not. Because if this is the DNA, a primer will be here, right? It will be complementary to the DNA. But adapters are not complementary, they will be added to the DNA.
oh, actually, means ligation kind of.
Yes, yes, yes. So why is that done? Because let's say you can have a lot of DNA, your samples might have anything, so the DNA sequence will change. But if your adapter sequence is known and you add your adapters to both the ends, so what you could do, you could create a probe which will have complementary sequence to your adapter, and this DNA will bind.
So আরেকবার আরেকবার But
যদি ক্রিয়েট করো ডিএনএ এর সাথে, তো তুমি অ্যাডাপ্টারের সিকোয়েন্সটা তুমি জানো, বিকজ অ্যাডাপ্টার তুমি বানিয়েছো।
হ্যাঁ
এবার অ্যাডাপ্টারের কমপ্লিমেন্টারি যদি আমি একটা প্রাইমার বা কিছু ডিজাইন করি, তো আমার যে ডিএনএ যাই থাকুক, কিন্তু ওটা বাইন্ড করে যাবে, বিকজ অ্যাডাপ্টারটা তো বাইন্ড আছে না ডিএনএ এর সাথে।
বাইন্ডটা কার সাথে করবে অ্যাডাপ্টার না ডিএনএ সাথে
অ্যাডাপ্টারের
Adapter
সাথেই
সাথে
করবে,
probe.
অ্যাডাপ্টারের সাথে
Okay.
করবে, তারপরে যে ডিএনএটা আছে সেটা যে কিছুই থাকতে পারে, সেটা পলিমারেজ কাজ করবে, তাই না? কিন্তু আমি প্রাইমারটা যদি অ্যাডাপ্টার হিসাবে রাখি, তো আমার প্রাইমার ডিজাইনিং এর স্টেপটা অ্যাটলিস্ট কমে যাচ্ছে। প্রত্যেকটা আলাদা আলাদা ডিএনএ এর জন্য আলাদা আলাদা প্রাইমার ডিজাইন করতে হচ্ছে না। আর ইউ আন্ডারস্ট্যান্ডিং দিস?
yes yes
So এখানে we are not doing primers. What we are doing here is there will be beads. Okay, so these beads will have the complementary sequence to the adapters that you have made. So and then there you will add PCR reagents like DNTPs, the polymerase, and all those kind of things will be there, and there will be an emulsion oil. This oil will be present so that there is only one bead in one oil droplet. Fine. And if your DNA has properly bind to that adapter, then what would happen? Polymerization will happen within this oil droplet.
so
Clear?
how can we make sure that there is only one bead in one oil droplet
Yes, sir. That is the question.
I mean, that is how these, I mean, the oils and PCR, I mean, the beads and the oils, those conformations are designed. So they have done a lot of studies. They have done optimization to make sure that this happens. But even if it doesn't happen, so what would happen? We'll come to the next step. So let's go there
and
and
also
then
so why is oil used sir can you explain one thing?
oil. What else? How could you create an emulsion in water if there is no oil?
No, why do we need it that's my question.
So if we have multiple beads,
Hmm.
and multiple beads are giving multiple, so there will be like we will see there will be pyro sequencing involved here. So if all the beads are together, beads might interact between themselves. A lot of things could happen. So we need to, keep them separate. So each bead will represent one reaction.
Means beads can be complementary to each other.
Yeah, it could happen, no, because they also have adapters. They could form aggregates also. Now, what lot of things could happen. So to make sure that your DNA binds properly with the bead, it is essential that you have this oil emulsion. Because in water, it will be difficult. I mean, the things get tricky. So let me see. What was I thinking? I was thinking about something here. I forgot about it. Let me see. What was I thinking about? Let me see. I think I'll remember it once we go forward. So essentially we see. Oh, droplet PCR. I was thinking about it. So you know about droplet PCR?
No, sir.
Droplet PCR, what we do, we create these small oil droplets. So there we amplify the PCR products only in those one droplet. So what it helps us identify how much PCR, I mean, let's say if we are doing RT-PCR or something like that. So, we need to take controls like the housekeeping genes. Anything? Does this ring a bell? Housekeeping gene, or
And
Yes,
naam
sir.
सुनती
We have learned about them. Housekeeping genes.
what are housekeeping genes?
Housekeeping genes are basically genes that serve the purpose of constitution in the cell itself.
That means they are always expressed.
Always expressed.
So these housekeeping genes are always taken as a control. So let's say I say that there is an overexpression of some particular, you know, protein. So there is a higher amount of transcript, mRNA transcript there. So I will always compare it with housekeeping genes, because whenever I'm doing extraction, it could happen that I simply have extracted more RNA. That's why I'm getting more less CT value. So in all the experiments, we take the control of a housekeeping gene so that we can normalize. We have already studied this in one of the papers earlier, where citrate synthase was
Sir,
a housekeeping gene.
sir, droplet PCR টা back to explain করবেন
And
last
droplet
number.
PCR is not important here. Droplets PCR is like, I'll tell you what that is. So housekeeping genes are important so that we can relatively quantify whether our gene of interest is more or less. So if, let's say, you have extracted more RNA or DNA, then you will have more amount of housekeeping genes. I mean, the transcript level will also be high.
Yes.
So that way you relatively quantify ki whether there is some overexpression or whether the expression level is same, or whether it has been, down regulated, expression has been down regulated. So when we do droplet PCR, we get absolute quantification. So what happens in one, they also have an emulsion, an oil emulsion. There only few DNA copies could get in, and each droplet will have a separate PCR reaction. So you can actually determine without using housekeeping genes, without using controls, that what is the number of, let's say, genes that you started, what DNA that you started with. Clear, not clear?
So can we use glycerin?
Partially clear.
What?
Can we use glycerin here?
Glycerin actually does not cause emulsion. It will just, it's heavy, so it will come down. Oil can only create an emulsion.
Okay.
Some colloidal solutions can create emulsion, like even the fats present in milk, they cause an emulsion. Milk is an emulsion, you know that, na? So
Yes, sir.
usually oils are used. Anyway, let's not get into it. So we will perform emulsion PCR, and each beads will be isolated. This isolation of each beads is possible because of that emulsion. Otherwise, if it is in water, it's very difficult to do that. So then we place it in picotiter plates, and the idea is each bead will only fit into one of the wells, and then we will perform pyrosequencing. So enzyme will be there. Enzyme means the polymerase enzyme, and then there will be pyrosequencing. So you know what is pyrosequencing? No,
No, sir.
I think not. So what happens? So this is how pyrosequencing works. So there is a primer and there is a polymerase which will keep on adding DNTPs, right? Clear?
Yes, sir.
So there will be one, let's say A is added. It does not, A is not the complementary sequence. A is added, so it doesn't bind. So there will be no luminescence here. Fine. Then I will use an A pyrase enzyme to remove the unused A's. Then I will add T and see if there is any luminescence. There is no luminescence. Then I will add G and I observe luminescence. That means G is the first sequence. Clear?
Okay, sir.
Then I add C. Then again I see luminescence. So the first sequence is G, then it is C. Let's say if that after that we have T, so we will do this cyclically. We'll again add A, there is no luminescence. Then in T we observe luminescence. So like this, we have pyrosequencing results.
So what is the utility of this apyrase enzyme?
Apyrase removes any inorganic phosphates and the unused DNTPs. So
Means
if you do
it
not
degrades
remove,
them.
it degrades it, it removes it from the reaction.
Okay, okay.
So what could be one potential problem of this?
So time taking.
Yeah, that is true, of course. But anyway, apart from that, we have
The
machines designed
process,
to,
the process will be costly, and
costly is
the,
true.
and one more thing, it since it removes the inorganic phosphates, so that will might cause a problem.
So
What
like
if we have
high
re-
energy consumption.
what if we have a lot of repeats? A stretch of A. Let's say we have that. What would happen? All the A's will bind together.
All these will bind, huh?
So the fluorescence signal will increase. So let's say if
Oh,
this is the fluorescence for 1A, this is the fluorescence for 2A's, this is the fluorescence of 3A's. So 1 থেকে 2 এর হয়তো there will be 2 times difference, but 2 থেকে 3 এর হয়তো অতটা difference থাকবে না because there is a detection limit also, no?
Yes, sir.
So when there are repeats, then it is very difficult to figure out what is happening. So how many repeats, I mean, how many A's are there or T's are there or G's are there? It's very difficult to figure that out.
Okay.
Clear?
Okay, sir.
Okay, so then we will come to third generation sequencing. So so far so good. There is a reaction where here just let me check. I must be having it here somewhere. I have missed the reaction, important reaction that you will also read. So yeah, so luciferin actually changes to oxyluciferin. That happens in presence of so ATP is required that will give light. So ATP will be converted to light. So the energy will be converted to light. So this will give us the fluorescence essentially. So this is how the reaction works, how the luminescence is produced. So that is how it works. So pyrosequencing means each of these, whenever there will be an incorporation of a base, what would happen?
There will be a fluorescence signal.
Extra phosphate group will be produced.
Extra, huh?
These phosphate group will be helpful in this reaction. So whenever this phosphate is produced, inorganic phosphate is produced, at that moment this reaction happens. So oxyluciferin happens and light is produced. Clear?
Yes, sir.
But if this inorganic phosphate will not be produced if the base is not added, clear?
Yes, sir.
So there will be no luminescence. Clear on that? Everybody is clear?
Yes, sir.
Anybody else?
So that means pyro sequencing and the previous one, that is the that PCR using this emulsion. These are next generation sequencing, right?
Yes, yes, yes.
Okay.
Here we perform the pyrosequencing reaction. So the
Yes, sir.
bases are added. As soon as it is added, there is a detector here which is monitoring all the fluorescence readouts. I mean, yeah, fluorescence readouts, and it is giving you a constant analysis of what base is added.
Okay. So means this total process is pyro sequencing, like from the beginning when we are
No, no, no.
adding the
Only whatever is happening here is pyrosequencing.
inside the
Feed inside the well.
and
So
what is
then
the
we
name
have
of the process? Name of the process first means previous to this pyro sequencing.
this is this one. This is next gen sequencing. We will see what examples we have. It's like Illumina sequencing. You have heard about it, Illumina, ion torrent sequencing.
Heard
These are all
heard about it, but don't know.
next gen sequencing. They use similar principles. Few of them have developed their own patents. All of them, they have their own patent, which have their own customized technology. Few of them, they use some other technologies also, but this is the basics. So we'll talk about third gen sequencing. So this is PacBio smart sequencing. So what is PacBio? Pacific Biosciences. This is a very famous company in the US. So here also with the sequence, we have adapter and they have this again patented polymerase, which will add fluorescent nucleotides. So here the DNTPs, they are fluorescent. So whenever this fluorescent nucleotides are added, the signal is recorded. And that signal will essentially give us what is the sequence. Clear?
So what's nano
Yes,
well?
sir.
What is?
Nano well.
Nano well is it's in the nanometer range. It's a very small well.
I mean, it's like the same principle of that well in last two slides.
Like picotiter plates we had,
Yes.
this is nano well. They have their own kits available for all this.
And so one thing,
Sir,
sir,
what
it
is this like patented polymerase? What did you say that? What is that thing?
I mean, I cannot tell you which polymerase this is because I don't know.
Okay, okay,
These
okay, no
these
problem.
kind of technologies are patented, so they get billions. They are selling. I'll tell you about Oxford Nanopore, how much the machine costs. So they are selling it for so much amount of money because they have these polymerases designed specifically in a way that they know it would work and it would give good results. So you cannot simply design this by yourself. That's what I'm trying to say. And even there is a detector also in each well. So that's the thing. Another thing, can you observe here?
yes, sir, those are adapters. Right?
Adapters are there. So what could essentially happen is there could be multiple readings also. Like sometimes there are like one end reading, there are paired end readings. So the benefit of third generation sequencing or even next generation sequencing is you have multiple readings. In Sanger sequencing, you have only one reading. So in multiple readings, the chance of error in reading is reduced. Clear? Okay.
I mean, those adapters are responsible for the paired reading.
See, it can circulate like this, no?
Okay, okay.
So that's the thing. And then this is Oxford nanopore sequencing. Here we have an helicase enzyme which unwinds the DNA and they will have the bases. No polymerase is involved here. So each base will give a separate, it will have a separate electronic, electric field associated with it, which will change the ion composition or the ions, how they behaved. Electrical signals will be changed based on the base. So this particular, each base will give a separate electrical output. Based on that, they decide whether which base is present. So if there are modified bases like methylated cytosine, will it give the same output as a cytosine?
No.
No.
So even methylated bases, they can also be checked using this kind of technology. So here also the helicase, it is patented. So they have optimized it. So this is how
Sir,
a
what is the meaning?
flow cell
What is
looks
the
like.
meaning of this line that the adapter molecule keeps DNA bases in place long enough to be identified electronically? Oh, what is the meaning? I mean, how is this adapter working here?
This is the adapter essentially. This is an enzyme that keeps the I mean, if the DNA is unwound and it is not going in a like a flow, then you cannot actually make out. If it again tries to make an helical structure, then you will not get a proper reading, no electrical reading. So it
Yes, sir.
makes sure that this is the adapter, that this DNA is straight and each and every base is read properly.
But how is it like ensuring this? I mean, the adapter is also a DNA molecule, right?
They have done no adapt. In this case, it's not. Here the adapter molecule is this protein. I mean, sometimes these kind of definitions they change. So what it contains, which kind of protein it contains, that is why they are taking so much money.
Okay.
So this is Oxford nanopore sequencing machine. This is essentially the gridiron is the machine. Any guesses how much does this cost?
Maybe 2 crores, maybe 2 crores,
2
3
crores
crores,
is too much, not that much.
50 lakhs.
Close 70 lakhs, around 70 lakhs it costs. We have this Oxford nanopore sequencing technology in our lab. So we have this
So
machine. This
in
is the
your lab,
CPU itself. Yes,
so in your lab or
yes.
our also.
No,
In
that is not present in our
were you
lab. Okay,
expecting
okay,
in
from the
heritage?
professional.
Yes, so for a second I thought, yeah.
No, 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 no. I wish that happens at some point. But anyway, this is how it looks like. This is the entire CPU also. I mean, this is the CPU, is a Linux-based system. And here we attach the flow cells like you have seen. So each flow cell costs around 2 lakh rupees, little less than that. So if there are 5 flow cells, it will be like how much? 50 lakhs? No, no, 10 lakhs, sorry. So it's like that. So it's a very, very expensive thing to do. So have you heard about sites like 23andMe, and there are something else also? I only remember this.
What's it?
23andMe. Then there is something else. I forgot the names. You know what it does?
What's it?
So you have to send in your sample, they will extract the DNA and they will perform this sequencing, and they will tell you what is your predisposition to certain types of cancers, any kind of diseases, what are your chances of getting a cardiovascular disease, and all these kind of things. Even they provide these services like if your family also has this 23andMe basically does sequencing. So if they have data for your family, they will create a tree kind of structure where you will get from which lineage you belong to and all these kind of things. So they cost around $1,000 for per person per sample, basically around 70-80,000 rupees. So you can get your DNA tested and do that probably. So this is essentially first generation sequencing, Sanger sequencing, Maxim, Gilbert, sequencing. So it is good for small DNA fragments. Second generation, as I said, Illumina or Ion Torrent, they are high throughput. I mean, they give faster result, more amount of DNA could be put, and you could get parallel reactions also. Here also the fragments are small. Third gen sequencing, it's almost the same as second gen sequencing, but the only difference is it can give you long reads. So what would be the advantage if we have long reads?
Higher accuracy
Not
means less
exactly. Not
less chance of
exactly.
error. I mean, like
No,
because
no,
we
no,
saw
no, actually not.
We can sequence the entire genome of some organisms
That
at
would be
once.
easier.
am I
Yes.
time saving kind of.
Time saving, yes. Another step, if you remember in the workflow, it was mapping with the reference genome. So when you create maps, so let's say this is the reference genome and you have a lot of fragments, let's say 1, 2, 3, like this, very small fragments. So mapping them will be very difficult. But if you have longer fragments, then there will be more overlaps. There will be easy. It will be easy to map. I mean, are you understanding this?
Yes, sir. Yes, sir.
Yes, sir. Global.
That way it is easier. So this is the overview of sequencing. If you see the market of Germany, this is a projected market. It is supposed to grow a lot. So it's already around $800 million in Germany alone. And the sequencing costs, they have progressively decreased, as we can see from this NIH data in the right. So that's all. I really hope you really use sequencing
Sir,
in one of your.
sir, can you please go to the slide of this packed bio smart sequencing once? Here, sir, in this process, uh, what are we actually? No, sorry, that that machine, that next slide, I guess, that where iron environment, Oxford Nanopore, sorry. Right. So in this system, this adapter molecule is the protein itself, which is actually keeping the DNA bases in place. Like they are maintaining the linearity and flow of the DNA bases so that we can detect the electronic signals. But sir, how are we like means for cytosine we have one electronic signal, for methylate cytosine we have another electronic signal. So how are these electronic signals actually like deciphered or derived? Can you tell
For
this
that,
means
there are
how
they have developed softwares for that.
like for
You know how mass spectroscopy result looks like?
no sir, no sir.
So if you see some result, it will be this, or FTIR results, they will show you like this, peaks will be shown. And each peak will correspond to some, if I talk about FTIR, each peak will correspond to some, let's say COO bond or CO bond, like that. So here also there will be separate electronic output to each of these bases, right? That is clear.
Yes sir.
So there will be an change. Let's say there will be an electrical charge which will be changed. So based which charge corresponds to which particular um, base, that they have developed their own softwares. So when they give you this machine, they also give their software to you, which does this
So
analyzing
that
thing.
means that is determined by this ionic environment inside this protein, adapter
Yes,
protein.
yes, yes, yes.
Okay, okay. Now means the total adapter protein is not responsible, but a specific portion is like a specific
Ions are there.
domain
Yes,
or
that
motif.
they. Hmm.
A specific domain or motif is responsible for detection of this electronic signal. So means as the basis will change, the ionic environment will also change.
Right, right, right. Absolutely.
Okay, okay. Means why and why, but why does that happen? Means how does that happen? Means suppose if adenine comes, suppose we cite an example. Suppose if A comes, then then due to the means is it due to the presence of those like substituent side groups like NH2 and nitrogen lone
Of
pairs for
course.
which
Yes,
the
yes. You're
ionic
right.
environment changes? But for that the adapter molecule like detects the presence of NH2 or something, and then it
Yes,
facilitates the ionic interaction, like it changes the
yes,
ionic
yes,
interaction
yes,
for which we get a different electronic signal, and then we
yes.
can actually detect that this is the okay. And sir, in the previous slide, in Spec Biosmart, okay, how are we getting this like loop of DNA means? Is it due to the adapter molecules?
Yes, adapters. They are complementary in such a way it will form a loop.
Okay, 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 sir, sir.
Yes, Shinjon, any
That means
questions?
So
that
one
means
question,
sir,
sir.
okay,
Let's
Yeah.
let him continue.
no, Sinjan, well.
I was like, in the second generation, you said like we have to make DNA library, right? So like, how is it done? It was I guess in slide 10.
This one.
Sorry, 12. Yeah. How is it done? Like we just add the
Adding the Yes, adding the adapters, preparing the DNA, you have to see whether your DNA is of good quality, whether your DNA is not with any proteins or something like that. This is what is DNA library preparation means. Okay,
like it does
let
not
me.
require any reference.
Reference is required in analysis.